viSNE is a tool for reducing high-parameter biological data down to 2 dimensions while still retaining single-cell resolution. This allows the researcher to not only visually identify phenotypically distinguishable cell subsets, but also gate these single-cell events. viSNE in Cytobank uses the Barnes-Hutt implementation of the t-SNE algorithm. This is an example of what viSNE data look like after an analysis. The result is a normal FCS file with 2 additional unique channels designated by the names t-SNE1 and t-SNE2. When visualized as the x and y axis of a traditional biaxial dot plot, a map emerges where each event is grouped among other similar events according to all the channels in the analysis. The grouping of events in viSNE is best illustrated by coloring the dots on the plot by an additional channel, which is a feature that has been added to traditional dot plots in Cytobank. Observe what happens when the map is colored by different channels. With each channel choice, a certain segregated area of the map lights up. The color corresponds to the intensity of the coloring channel in those events. With this view, all the events in the experiment are present. Global patterns can be observed easily, along with single-cell details. To begin a viSNE analysis, navigate to the Experiment Details page of an experiment. Click the link for Create viSNE Analysis on the left of the page. Enter a name for the analysis, and click OK. When the viSNE Setup page appears, it will list all the populations created from gating already done in this experiment. If gating still needs to be done, simply click the gating link to the right. When a population is clicked, a list of files will be presented. For simplicity, this experiment only has one file. When a population and a file combination has been made, the total number of events available from it will be listed. The number of events that will actually be sampled is based on the target specified below. When choosing to sample from multiple populations, the role of proportional and equal sampling becomes clear. If 2 populations of differing abundance are chosen, proportional sampling conserves that relative abundance. Toggling equal mode, however, forces equal sampling across all population file combinations. The channels selected for viSNE provide the criteria by which viSNE gauges similarity between events in order to group them. The channels selected should contribute to the separation of the desired populations. Avoid mixing linearly and non-linearly scaled channels in the same viSNE analysis. Compensation can be selected here for fluorescence experiments. Data scaling is important to any analysis, and viSNE is no exception. This area serves as a reminder, and a convenient link to the gating interface where the quality of scaling can be assessed. The only factor that affects viSNE is the scale argument of data transformed by the hyperbolic arcsine equation. When the viSNE analysis is started, a progress bar will appear. During this time, the run is actually being exported to a different resource for dedicated computation. You can leave the page or close your browser. When the analysis is finished, a link to the results will appear here, as well as on the Experiment Details page in the Advanced Analyses box. When the viSNE analysis has completed, the screen will automatically navigate to the gating interface with the t-SNE channel selected and with the plot type as colored dot plot. The gates from the original experiment will carry across, as well as any compensations and annotations. To compare the quality of the original gating to the separation of populations by viSNE, try changing the active population. This is the population as defined in the original data being displayed on the viSNE map. Seeing extra events scattered around the viSNE map indicates nonspecific gating, as seen here. The data can be gated per typical workflow. Here it is demonstrated how CD4 positive cells might be gated. Fast forwarding shows more gates. Click Apply and Return to go back to the illustration. Useful figures to explore viSNE data can be made in the working illustration. Here is an example of visualizing plots on a variable coloring channel defined by the channel's figure dimension, while the X and Y channels are defined statically as the t-SNE channels. The data can be scanned quickly to see expression levels of different markers across the viSNE map. There are different configuration options available for the dot plots colored by channel. Back in the gating interface is a convenient location to demonstrate them, though dot plots can be configured in the same fashion in the working illustration or in the gating interface. The dot plotting method has 2 options, binned pixels and stacked dots. In binned pixel mode, collisions between events in viSNE space are binned into single pixels on your screen, and then that pixel reports the median value of the events inside of it on the coloring channel in question. The stacked dot option arranges the events without binning, on top of each other, depending on the order in which they appear in the FCS file. This feature is useful for areas of data that are sparse in order to increase visual quality. For example purposes, I've gated the data on the time channel in order to reduce it. Binned pixels can leave gaps in the visual cues, which stacked dots can help fill in. The dot size and shape can also be configured. Rings are hollow in the middle and can help visualize events that might be overlapping because of stacking. The coloring channel range is the minimum and maximum values of the color bar, which is shown to the right of any dot plot colored by channel. The color bar is the key for associating a color in the plot to a particular value for expression intensity. The minimum and maximum values of the color bar are decided depending on the selected method in the menu. Global mode sets the boundaries of the color channel based on information from all files in the experiment. Local mode sets it based on individual files. Since there is only one file in this experiment, local is equal to global. User-defined mode allows a custom range. For more detailed information about calculating the color range, consult the pop-out help box. This concludes the introductory video on viSNE. The data shown are courtesy of the Irish Lab at Vanderbilt University and are publicly available on the Fluidigm Cytobank server at the address shown. For questions, feel free to email helpdesk@cytobank.org, visit our live support chat, or consult our knowledge base or the viSNE FAQ. Thank you.